Today's video is about the question of what are cell surface markers and what can the potential application of cell surface markers be. So the simple explanation of cell surface markers is that they are molecules on the surface, basically the external surface of a cell, that mark a specific cell type. Now, the real life example that comes to my mind that can be used for describing a cell surface marker is that markers are arranged on the surface of the cell like tall lamps on city roads, which is shown on the left side of the slide. However, a cell is three-dimensional, and therefore the arrangement of markers on the surface of the cell are shown on the right hand side like thorns are arranged on cacti. So on the next slide is an example of certain type of cells of the immune system called B cells. They have markers called immunoglobulin on their surface. On the left side of the slide are these Y-shaped molecules called immunoglobulins. And on the right side of the slide is shown these immunoglobulins, which are on the surface of these round-shaped cells. So, as mentioned earlier, the question was, what can be the potential use of cell surface markers? Well, in cancer, it is thought that most probably the seeds of cancer are a particular type of cells called cancer stem cells. Now, these cancer stem cells have specific markers on their surface called CD markers, where CD stands for cluster of differentiation. As you can see in the photograph over here, this cancer stem cell, in this particular case a liver cancer stem cell, has markers such as CD133 and CD90 on the surface of the cell. So these markers can be used as targets for drugging the cancer stem cell as shown in the next slide. For example, there are discoveries which have been made in various pharmaceutical labs called drug antibody conjugates. In such a case, as you can see on the right hand side of the slide, a drug is bound to an antibody which is against these CD markers on the cancer stem cell. So when the drug antibody conjugate binds to the CD marker on the surface of a cancer stem cell and then they get internalized by the cell, then this particular drug, which is cytotoxic or cell killing, is released inside the cell and kills the cancer stem cells. So in this way today we discovered these unique molecules, the mark a specific cell type, and how discovery of these unique molecules can make them attractive drug targets, especially in diseases such as cancer. I hope you enjoyed this video.